Hey guys, welcome back to another episode of the USMLE Guys. My name is Dr. Paul. Today we're doing another high-yield USMLE Step 1 Q&A. Now, this lecture is based on our Step 1 crash course, which is a high-yield crash course that asks you questions based on material outlined in the first aid. So if you enjoy this and you find it to be useful, you can check out the full crash course in the link description below. The specific topic we're going to discuss in this lecture is metabolism. Let's not waste any more time. Let's dive in with today's Q&A. All right, guys, welcome back to the next lecture. Now, we're about to start the metabolism section. Now, what I want to do with metabolism here is sort of stick to the bigger picture, because going into the nitty-gritty details, every single step of every single pathway makes no sense, especially for our purposes, because that's more of an undergraduate thing to know every single step. What we need to know are the rate-limiting steps, energy production, energy usage, pathologies associated with pathways, etc. So when we dive into this stuff, if you're not familiar or you need to review this stuff in more detail, I recommend that you get one of your biochemistry textbooks or go through your class notes and dive deeper. For our purposes, though, we are going to stick to basically what the first aid outlines and go into just some superficial detail, not detail, but superficially, we're going to sort of go over what we need to know in enough detail so that we touch on the important stuff, but not so much that we're touching on things that are unlikely to be tested, because ultimately this crash course is designed to just help you get through everything, see it again. Hopefully it's either if it's your first time going through everything, then it's exposing you to the important stuff. And if this is your last time going through everything, it's a way for you to do a review of things you should know. So with that said, I just wanted to clarify that. Let's dive in and let's get started with our first question, which is a multiple choice. So go ahead and hit that pause button, figure this one out, and then come on back when you think you have the right answer. The correct answer here is D. So this is on the surface super easy, but it's a commonly tested concept and you absolutely must know it 100%. So I wanted to make sure that you remember this. So don't forget, the processes that happen in the mitochondria include fatty acid oxidation, the TCA cycle, the production of acetyl-CoA, oxidative phosphorylation, and ketogenesis. The processes that happen in the cytoplasm are glycolysis, the HMP shunt, and the synthesis of nucleotides, fatty acids, proteins, and cholesterol. And last but not least, the metabolic processes that happen in both the cytoplasm and the mitochondria. High yield stuff, you need to know that. The urea cycle, gluconeogenesis, and heme synthesis. All right, let's move on to the next question. We have a matching exercise. So what I want you to do is match the process with its rate-limiting enzyme / rate-limiting step. Figure this one out, come on back, and then we'll discuss all of the correct information. All right, so here are the correct answers. If you need to fix anything, go ahead and do so. Otherwise, let's start at the top here with glycolysis. This is, of course, regulated by PFK1. And don't forget our activators and our inhibitors. So remember, both AMP and fructose 2,6-bisphosphate will activate this, while ATP and citrate will inhibit it. Now, gluconeogenesis is regulated by fructose 1,6-bisphosphatase 1 and is only going to be inhibited by the presence of either AMP or fructose 2,6-bisphosphate. The TCA cycle is, of course, regulated by isocitrate dehydrogenase. It is activated by ADP and inhibited by both ATP and NADH. Glycogenesis is regulated by the glycogen synthase enzyme and will be activated by three things: glucose 6-phosphate, insulin, and cortisol, and will be inhibited by two things: glucagon and epinephrine. Now, glycogenolysis is regulated by glycogen phosphorylase, and this is activated by three things: epinephrine, glucagon, and AMP, and inhibited also by three things: glucose 6-phosphate, insulin, and ATP. Now, the HMP shunt is regulated by the glucose 6-phosphate dehydrogenase enzyme, very important enzyme, we'll get into that later, and this pathway is activated by NADP+ and inhibited by NADPH. And don't forget that this enzyme is deficient in the glucose 6-phosphate dehydrogenase deficiency. That is inherited how? X-linked recessive. The urea cycle is next. This is regulated by the carbamoyl phosphate synthetase 1 enzyme, and its only regulating factor is N-acetylglutamate, which is an activator. Fatty acid synthesis is, of course, regulated by the acetyl-CoA carboxylase enzyme, and the synthesis of fatty acids will be activated by the presence of insulin and citrate, and it can be inhibited by the presence of glucagon or palmitoyl-CoA. Fatty acid oxidation, on the other hand, is regulated by the carnitine acyltransferase 1 enzyme, and that's inhibited by the presence of malonyl-CoA. Ketogenesis is regulated by the HMG-CoA synthase enzyme and has no regulating factors, while cholesterol synthesis is regulated by HMG-CoA reductase. And this is activated by the presence of either insulin, thyroxine, or estrogen, and inhibited by the presence of either cholesterol, obviously it's cholesterol, and glucagon. Now the final two pathways we have are de novo purine and de novo pyrimidine synthesis. Now de novo purine synthesis is regulated by the glutamine PRPP aminotransferase enzyme and is only inhibited by the presence of either IMP, AMP, or GMP. And the last one is de novo pyrimidine synthesis. This is regulated by the carbamoyl phosphate synthetase 2 enzyme, and this is activated by ATP or PRPP and inhibited by UTP. All right, let's move on to the next question. So we have another matching exercise here. So what I want you to do is hit the pause button. I want you to match the features of hexokinase and glucokinase with the correct enzyme. So go ahead and hit the pause button, figure this out, come on back, and we'll discuss the differences and similarities and what you need to know about hexokinase and glucokinase. So here are your correct answers. If you need to pause and fix anything, please go ahead and do so. Otherwise, knowing the differences between glucokinase and hexokinase will almost certainly get you a point on your exam in some way, because this seems to either always pop up on a question that will require you to be able to pull on this information, or they might just directly ask you about the features, such as this matching exercise. So either way, it's important to know this because it will in some way, shape, or form help you answer something on exam day. So let's take a look at the differences between the two. So remember that in the liver and the pancreatic beta cells, glucokinase will be used to catalyze the phosphorylation of glucose into glucose 6-phosphate. Hexokinase is going to do it everywhere else. Now, the exception to hexokinase is, of course, the liver and the beta cells of the pancreas, because that is where glucokinase is used. Now remember that hexokinase is going to be essential for gathering glucose in the tissues for use when glucose levels are low. That will be important to remember momentarily. Now, when glucose levels are high, the liver uses glucokinase, and that helps us to store glucose. Now, for those two reasons I just mentioned and told you to remember, that is why the Km of hexokinase is low, which means that it has a higher affinity for glucose, while the opposite is true for glucokinase, where the affinity is lower because glucose is more abundant. Now, this makes sense because, remember, glucokinase is used when glucose is abundant and we want to actually take it and store it. Now, Vmax is also lower in hexokinase as opposed to glucokinase. Now, since glucokinase is used when glucose is abundant, do you think that it is induced or inhibited by insulin? Think about it. It will be induced by insulin, right? Hexokinase won't, because glucokinase is used when glucose is abundant. When glucose is abundant, insulin levels increase. And finally, hexokinase will receive feedback inhibition via the glucose 6-phosphate enzyme, while feedback inhibition of glucokinase will be with the fructose 6-phosphate enzyme. All right? Basic stuff, but important stuff, so make sure you know that. All right, let's move on to the next question. Go ahead and hit that pause button, figure this one out, and then come on back when you think you have the right answer. The correct answer to this question is C. Now, remember in undergrad how we had to know every single step of glycolysis and all the other pathways? Well, as I said earlier, on the USMLE, you should focus your attention on knowing which steps produce and use ATP, as well as any pathologies or special products that might be made. So in glycolysis, remember that we've got two steps that require ATP, which are when we go from glucose to glucose 6-phosphate, which, don't forget, we'll use either glucokinase or hexokinase, and the step that converts fructose 6-phosphate into fructose 1,6 bisphosphate. And remember, That is a step using our rate limiting enzyme, which is what? PFK1. Now, do you remember the two activators and the two inhibitors of the PFK1 enzyme? Our activators are AMP and fructose 2,6-bisphosphate. Our inhibitors are ATP and citrate. We've also got two steps that produce ATP. Those are the steps whereby we convert 1,3-bisphosphoglycerate into 3-phosphoglycerate, and that's done via the phosphoglycerate kinase enzyme. And then the final step where we take phosphenolpyruvate, PEP, we turn it into pyruvate. That, of course, happens with the pyruvate kinase enzyme. Don't forget about the two important enzymes, fructose bisphosphatase 2 and PFK2, which are bifunctional enzymes that are actually the same, and which is present, and which would be present would depend on whether we're in the fasting state or whether we're in the fed state. So if we're in the fed state, which we can think of under normal circumstances when looking at glycolysis, the enzyme PFK2 will convert fructose 6-phosphate into fructose 2,6-bisphosphate, which is, of course, going to be an activator of that rate-limiting step involving PFK1. Now, if we're in the fasting state, fructose bisphosphatase 2 is active, and it reverses things, converting fructose 2,6-bisphosphate into fructose 6-phosphate. That then goes on to the production of glucose via gluconeogenesis. It's also important that you know how the fed versus fasting state will cause each of these to become active. So if we're in the fed state, when insulin rises in response to increased glucose, the cyclic AMP level lowers, which lowers pKa, which decreases fructose bisphosphatase 2. This increases PFK2. That results in the progression of glycolysis and the eventual production of ATP. Now, on the other hand, if we're in a fasted state, a rise in glucagon happens, increasing CAMP, that increases pKa, which increases the expression of the fructose bisphosphatase 2 enzyme, which lowers PFK2. That shunts things away from glycolysis and then towards gluconeogenesis. Now, if we successfully make it to the end of glycolysis, we get pyruvate. That needs to go through the pyruvate dehydrogenase complex so that we can move into the TCA cycle. It's that complex bridges the gap between glycolysis and the TCA cycle. So remember, this complex will consist of 3 enzymes that require 5 cofactors. Those cofactors include thiamine pyrophosphate, lipoic acid, CoA, FAD, and NAD+. An increased NAD+ to NADH ratio, as well as an increased ADP to calcium ratio, will activate this complex. Now, there's an X-linked disorder associated with this complex known as the pyruvate dehydrogenase complex deficiency. And in this condition, we will see that pyruvate builds up because it can't move forward, and then it gets shunted into either the production of lactate via LDH or into alanine via ALT. Now, this condition is characterized by lactic acidosis, of course, lactate's increasing, neurodefects, and an increase in serum alanine that is seen during infancy. Now, this can be managed by increasing the intake of certain nutrients, specifically ketogenic nutrients. All right, let's move on to the next question. We've got a multiple choice here, so go ahead and hit that pause button, figure this one out, and then come on back when you think you have the right answer. The correct answer here is E. So remember that the fate of pyruvate is to either be converted into alanine via ALT or lactate via LDH, both of which occur in the cytosol, or into oxaloacetate via the pyruvate carboxylase enzyme, or into acetyl-CoA via the pyruvate dehydrogenase enzyme. Both of those steps would occur in the mitochondria. Now remember that converting pyruvate into lactate uses NADH, while converting pyruvate into acetyl-CoA makes NADH. And converting pyruvate into oxaloacetate requires CO2, and it requires ATP. Okay, don't forget that. All right, let's move on to the next question. We have a multiple choice here, so go ahead and hit that pause button, figure this one out, and then come on back when you think you have the right answer. All right, the correct answer here is E. So once pyruvate is converted into acetyl-CoA, it's going to enter that TCA cycle. Now remember that our rate-limiting enzyme here is isocitrate dehydrogenase, and that we have a few enzymes in this cycle that are irreversible. That includes the rate-limiting step or the rate-limiting enzyme, alpha-ketoglutarate dehydrogenase. That converts, of course, alpha-ketoglutarate into succinyl-CoA, and citrate synthase, which converts oxaloacetate and acetyl-CoA into citrate. There's a few important key points that I want you to remember about energy production and the TCA cycle. So First of all, remember that the process of converting a pyruvate into an acetyl-CoA gives us one CO2 and one NADH molecule. Every single acetyl-CoA that enters into that cycle will produce the following: 1 GTP, 3 NADH, 1 FADH2, and 2 CO2s. Overall, this is worth a total of 10 ATPs. Now, the AKG dehydrogenase enzyme, alpha-ketoglutarate, which I mentioned converts alpha-ketoglutarate into succinyl-CoA, it needs the same cofactors to work as the pyruvate dehydrogenase complex does, meaning it needs a lot. So of course, don't forget, those are lipoic acid, vitamins B1, vitamin B2, vitamin B3, and vitamin B5. Okay, it's really important that you remember what's made in this cycle. All right, let's move on to the next question. Multiple choice. As always, go ahead and hit that pause button, figure this one out, and then come on back when you think you have the right answer. All right, the correct answer is B. Now, when it comes to the electron transport chain, make sure you understand the big picture, which is, of course, that we take NADH or other hydrogen ions and donate those electrons to create a proton gradient. And that is going to be coupled to oxidative phosphorylation to help propel the production of energy. Now, complex 1 receives its hydrogen from NADH, complex 2 from FADH2. Ubiquinone is a compound that connects the first and the second complexes to the third. And as it gets reduced, it delivers electrons to that complex. So the third complex, of course, is also known as cytochrome oxidoreductase. And this consists of cytochrome B, a protein made of iron and sulfur, FeS protein, cytochrome C proteins, and a Reski center, which is a 2 iron, 2 sulfur center. Now, that'll come into play shortly. Complex 4 is composed of cytochrome proteins A, A3, and C. Now, this complex contains 2 heme groups and 3 copper ions. These cytochromes hold an oxygen molecule tightly between iron and copper until the oxygen gets reduced. Now, the gradient created up to this point allows for ATP synthase to catalyze the synthesis of ATP from ADP and, of course, phosphate. Now, some of the most common questions you will get with the ETC are the things that can poison it. So let's take a look. So complex 1 can be poisoned by rotenone. And this works by preventing the transfer of electrons from complex 1 to ubiquinone, and it does this by blocking the ubiquinone binding site. At complex 3, we have antimycin A, which binds to the QI site of cytochrome C reductase. That inhibits the oxidation of ubiquinol. At complex 4, we can see poisoning by cyanide, by carbon monoxide, hydrogen sulfide, and azide. And what these do is they bind more strongly to the oxygen center of that Fe copper center in cytochrome C oxidase that I mentioned, and that prevents the reduction of oxygen. And then at complex 5, or the ATP synthase, we have oligomycin, and that can inhibit ATP synthase by blocking the flow of protons through the subunit. We also have uncoupling agents. Now, these hurt the ETC by increasing the permeability of the membrane, and that will decrease the proton gradient, which allows the transport of electrons to continue, but uncouples it from ATP synthesis. Now, remember, these uncoupling agents are heat producers. And the big ones you want to remember are aspirin, brown fat, as well as 2,4-dinitrophenol. Okay, something on there will be tested, guaranteed. Make sure you know that stuff. All right, let's move on to the next question. Go ahead and hit that pause button, figure this one out, and then come on back when you think you have the right answer. The correct answer here is A. So let's take a look at gluconeogenesis, specifically focusing on the irreversible enzymes and their roles in this process. So first is pyruvate carboxylase. This is found in the mitochondria and is needed to convert pyruvate into oxaloacetate. And this step requires ATP and biotin as a cofactor. Now, do you remember what activates this enzyme? That would be acetyl-CoA. Now, PEP carboxykinase is next. PEP is phosphenolpyruvate. And this enzyme is found in the cytosol, and it converts oxaloacetate into phosphenolpyruvate. This step requires GTP. Fructose 1,6-bisphosphatase is another cytosolic enzyme, and its job is to convert fructose 1,6-bisphosphate into fructose 6-phosphate. Now remember from earlier that this is activated by what? By citrate, and inhibited by what? Two things, AMP and fructose 2,6-bisphosphate. Now, your final irreversible gluconeogenic enzyme is glucose 6-phosphatase. That converts glucose 6-phosphate into what? Into glucose. All right, let's move on to the next question. All right, another multiple choice question. So as always, go ahead and hit that pause button, figure this one out, and then come on back when you think you have the right answer. The correct answer here is C. So let's talk about the HMP shunt, which is needed to create NADPH and ribose. Now that NADPH is needed for reduction reactions, cholesterol synthesis, and fatty acid synthesis, whereas the ribose is needed for nucleotide synthesis. Now there's two phases in this pathway. We have the oxidative and the non-oxidative phases or reactions. Now don't forget which of these is irreversible. That would be the oxidative portion. Now which of these happens in the cytoplasm? That's a trick question. They both do. Don't forget that. Now in the oxidative reaction, glucose 6-phosphate is converted into 6-phosphogluconate. That creates an NADPH molecule in the process. Then 6-phosphoglycerate would be converted into ribulose 5-phosphate, again creating an NADPH molecule as well as a CO2. Now as a whole, this phase gives us 2 NADPH molecules, 1 CO2, and it uses 1 molecule of water. Now once we've got our NADPH, we're going to look into that non-oxidative phase of the shunt, which, don't forget, is going to be a reversible phase. Now the ribulose 5-phosphate that we made in the oxidative phase, that can be converted into ribose 5-phosphate via the phosphopentose isomerase enzyme. Now this ribose 5-phosphate can then go on to create whatever we need. So for example, it can simply go to aid in nucleotide synthesis, or it can be converted via transketolase to either fructose 6-phosphate or glyceraldehyde 3-phosphate. Now, there's a really important enzyme in the HMP shunt that's always tested due to its genetic associated condition. And I mentioned this earlier. What we're talking about here, of course, is the glucose 6-phosphate dehydrogenase deficiency. This is inherited how? X-linked recessive. So remember, when we're deficient in this enzyme, we can't make NADPH. As a result, we have an inability to reduce glutathione, and subsequently, we put red blood cells at an increased risk of being damaged by free radicals. Now, this results in red blood cells being susceptible to oxidizing agents, medication, certain foods. What's a classic food? Fava beans. Now, the consequence of this ultimately is going to be breakdown of that red blood cell, hemolytic anemia. Now, do you remember when we're dealing with hemolytic anemia, what types of bodies and which unique types of cells we will see? We'll see Heinz bodies, which, remember, these are seen as a result of denatured globin chains precipitating within the red blood cell, and we will see Bite cells. Bite cells are the result of phagocytic removal of those Heinz bodies by your splenic macrophages. Now, just to wrap this up, do you remember which drugs are commonly known to precipitate hemolytic anemia in a case of glucose 6-phosphate dehydrogenase deficiency? Think anti-TB drugs, sulfonamides, nitrofurantoin, as well as primaquine or chloroquine. All right, next question. We got a multiple choice. As always, hit the pause button, figure this one out, and then come on back when you think you have the right answer. All right, the correct answer here is D. Let's talk about disorders of both fructose and galactose metabolism. So first up, we have essential fructosuria. This is inherited in an autosomal recessive manner, and this is due to a deficiency of the fructokinase enzyme. Now, this is benign and typically asymptomatic, and it's characterized by the presence of fructose in the blood and the urine. Now, like I said, this is going to be asymptomatic, and the reason why is because hexokinase can just pick up the slack here and convert fructose into fructose 6-phosphate. Now, a more worrisome fructose-related condition is hereditary fructose intolerance. That is also inherited in an AR manner, but the deficiency is different. Here, it's caused by a deficiency of the aldolase B enzyme. As a result, we will see an accumulation of fructose 1-phosphate. That lowers the availability of phosphate and subsequently will inhibit gluconeogenesis and glycogenolysis. Now, the way you're going to be able to identify this is by the onset of symptoms after the patient eats anything that's rich in fructose, like fruit juice, or even honey. Now, don't forget that if they tell you in a vignette that the urine dipstick is negative, this is only because the urine dipstick is testing for glucose, not fructose. So it's kind of an irrelevant distractor. Now, since this is concerning, what symptoms do we need to look out for in a vignette? Well, I want you to keep an eye out for the symptoms of hypoglycemia, as well as vomiting. Some more severe and serious findings like jaundice and cirrhosis may also be seen. Now, in order to manage this, we need to decrease the patient's fructose intake, as well as avoid anything that can be converted into fructose, like sorbitol or anything that contains fructose, such as sucrose. Okay? Moving on to our disorders of galactose metabolism, we've got the galactokinase deficiency. This is also inherited in an AR manner, and this is caused by deficiency of the galactokinase enzyme. Now, as a result of an enzyme deficiency consisting of galactokinase, what's going to happen as a result is we will get an accumulation of galactocol if the patient is eating anything that contains galactose. Now, this is relatively benign, but it can result in infantile cataracts as well as an inability to develop a social smile or track objects. Next up, we have classic galactosemia. This is also inherited in an AR manner, and this is caused by an absence of the galactose-1-phosphate uridyl-transferase enzyme. This causes an accumulation of toxic galactocol, especially in the lens of the eye. Now, you'll typically see this starting when the baby begins to feed. And the reason why is because we have lactose in both breast milk and formula, and lactose contains both glucose and galactose. Now, the characteristics of this condition are failure to thrive, jaundice, hepatomegaly, intellectual disability, and infantile cataracts. Now, an important association to keep in mind with this condition is the increased risk of E. coli-related sepsis in neonates. And the way you're going to treat this is by excluding both galactose and lactose from the diet. Now, the enzyme that converts galactose to galactocol, which is aldose reductase, is also responsible for trapping glucose in the cells by converting glucose into sorbitol. Now, certain tissues can take this sorbitol and convert it to fructose via sorbitol dehydrogenase, but some tissues have an insufficient amount of that enzyme, and that will result in trapping of sorbitol intracellularly, and its accumulation can cause osmotic damage, which can manifest in ways like developing retinopathy, cataracts, peripheral neuropathy. Okay, and don't forget that the retina, the kidneys, and the Schwann cells, they have aldolase reductase, but they lack sorbitol dehydrogenase, while the lens has primarily aldose reductase. Now, some structures like the liver, the ovaries, and the seminal vesicles have both of these enzymes. Lactase deficiency is also something you want to keep an eye out for, especially as someone gets older, because it leads to lactose intolerance. Now, an age-related decline in lactase is known as primary lactase deficiency, while a secondary cause would occur when the intestinal brush border becomes damaged, because that's where lactase turns lactose into glucose and galactose. Now, certain illnesses like autoimmune diseases or gastroenteritis can be responsible for causing a secondary lactase deficiency. Now, someone with a lactase deficiency, or they would say I'm lactose intolerant, would have flatulence, bloating, cramping, and potentially even osmotic diarrhea. The way to fix this is quite easy. You have two options. A, you avoid foods with lactose, or you start taking lactase pills. Your choice depends how much you like milk products. All right, let's move on to the next question. It's a multiple choice. As always, hit the pause button, figure this one out, and then come on back when you think you have the right answer. All right, the correct answer here is D. So when it comes to the urea cycle, you need to remember a few important things. First, this one happens in both the mitochondria and in the cytoplasm. And even more importantly, remember that the rate-limiting step happens with the carbamoyl phosphate synthetase 1 enzyme and is activated by N-acetylglutamate, and that this specific rate-limiting step happens in the mitochondria. Now, the production of carbamoyl phosphate from that rate-limiting step combines with ornithine and is converted via the ornithine transcarbamylase enzyme into citrulline, which then makes its way into the cytoplasm. Now, citrulline will then require aspartate and ATP to create arginosuccinate, and with the help of the arginosuccinate synthetase enzyme, creates arginosuccinate. Now from here, arginosuccinate lyase creates arginine and fumarate. Then arginine is combined with water and via arginase makes ornithine and urea as a byproduct. Now, a deficiency of that rate-limiting ornithine transcarbamylase enzyme will lead to a buildup of ammonia in the blood. And this enzyme deficiency is inherited in an X-linked recessive manner, and it results in a variety of consequences like lethargy, ataxia, asterixis, slurring of the speech, and even death. Now, this is your most common urea cycle disorder, meaning it is most likely to be tested. And while it's inherited in an X-linked recessive manner, the other disorders of the urea cycle, they're all inherited in an autosomal recessive manner. So that's something to really keep in mind if you get a genetics-related question on your exam. You don't want to fall for this trap. So just remember that the most common one is the only one that's inherited, not in an AR manner, but it's X-linked recessive. Now, due to the increased levels of orotic acid that build up when this enzyme is deficient, you'll be able to find increased levels in the blood and in the urine. As well, you'll find a decrease in BUN and the symptoms of hyperammonemia, of course. Now, one of the ways by which you can differentiate this from orotic aciduria is by the fact that this does not cause megaloblastic anemia, while orotic aciduria does. Now, when ammonia levels increase, it's going to result in a change to the proportions to alpha-ketoglutarate, glutamate, GABA, and glutamine. And a decrease in those proportions will result in CNS toxicity and cerebral edema. That is as a result of the glutamine-induced osmotic shift that ensues. Now, the way hyperammonemia is managed is by limiting protein intake, as well as by giving certain medications that can actually lower the levels of ammonia in the body, such as lactulose. You can give certain antibiotics, as well as drugs that can react with glycine or glutamine to form products that can then be easily excreted by the kidneys. Lactulose, which I just mentioned, is a commonly tested medication. And if you're asked about this, the way it works is by acidifying the GI tract, which then traps those ammonium NH4 molecules so they can be easily excreted. Okay, so if you're asked that question, keep that one in mind. I've seen this one pop up a lot over the years. All right, let's move on to the next question. We have a matching exercise, so go ahead and hit that pause button, figure all of this out, and then come on back when you think you have the right answers. All right, here are all of the correct answers. If you need to fix anything, pause and go ahead and do so. Now, I'm not going to walk you through all this because it's right there in your books for you to see, but you want to make sure you know what creates what, because by knowing this information, it'll help you to know those foundational aspects of more complex and challenging questions. So for example, let's say you've got a question pointing to PKU and you're excited because you know everything about PKU, like the signs, the symptoms, the enzyme missing, etc. Well, what if they take it right back to the very basics and asked you about the substrates that will accumulate and that as a result will fail to make certain products? If you don't know that phenylalanine is the substrate to DOPA, dopamine, norepinephrine, and epinephrine, you'll miss out on an easy point. So please make sure you know these pathways, all right? It's right there in your books. All right, let's take a break right there. I'll see you guys on the next lecture. Thanks for sticking around to the end. I hope you found that to be useful and helpful. If you did, do me a huge favor, hit that thumbs up button below. Share this with your friends. If you're not yet subscribed and you want me to let you know every time we release brand new episodes, hit that subscribe button below, set up notifications, and that's exactly what I'll do. If you are interested in the full Step 1 crash course, make sure you use the link in the description below so that you can learn more about that. We'll see you guys on the next episode.